Yes, thank you for the organisers for inviting me to present as well. Today I'm going to be looking at perhaps a much drier topic than some of the other speakers have spoken about today. I'm unfortunately not covering any of the science, but the regulation of advanced therapies in Europe, and I'll be talking about how they're defined, the current landscape, so who's developing and what products are in the pipeline. Some of the key technical considerations, particularly when you're progressing to a first-in-man study. And then in terms of sponsors, particularly early-stage sponsors, what regulatory procedures are available in Europe to try and promote development. And finally, some avenues to increase efficiency to both the clinic and the market. So in Europe, the ATMP regulation was brought into place in 2007 and was implemented firstly in 2009. And really this was the first regulation in Europe which considered cell-based products and gene therapies as medicinal products, and therefore regulated under the Medicinal Products Directive. So the classification of these products takes into account their mechanism of action, the method of manufacture, as well as the nature of the active substance and the proposed use. And they broadly fit into these three categories of the gene therapy, somatic cell therapy, or, oh, I've got that twice. Sorry, that last one should read tissue engineering products. So the key questions, and really, again, this is all very dry, but it really defines how your product is interpreted by the regulatory agencies and what sort of data expectations that they will have for your product come at the time of assessment of a clinical trial application and a marketing authorization application. So for a gene therapy, it's relatively straightforward. Have you got a recombinant nucleic acid sequence involved? And does the primary effect directly relate to the expression of the nucleic acid, or to that nucleic acid sequence being incorporated into the human genome? In the case of a cell therapy or a tissue engineering product, the key questions are those cells or tissue substantially manipulated? And I'll come to that a bit later. The second question is the product intended for non-homologous use? So this essentially relates back to what the regulatory agencies talk about under the context of the same essential function. So in the case of peripheral blood or bone marrow cells that are being used for hematopoietic reconstitution or immune reconstitution, that would be considered as the same essential function and therefore homologous use. Whereas if you were using a mesenchymal stem cell product for wound healing away from the traditional site of effect of MSCs, then that would be counted as a cell-based product. And of course, the final criteria is that it's used for the treatment or prevention of disease. So, as I said, in the context of cell and tissue engineering products, one of the key issues in the classification of whether something falls into the regulation as a medicinal product or as a cell or tissue-based product is whether it's considered by the agencies to be substantially manipulated. One of the key ones, of course, is that if you do any kind of expansion in the dish, that's considered a substantial manipulation, because that, of course, increases the risk potentially that you're introducing certain selection of your cells in culture, and therefore that the agencies see that associated with needing more scrutiny, regulatory scrutiny. Examples of non-substantial manipulation would be things like basic freezing, cell separation, concentration or purification, centrifugation and lyophilization, fairly standard procedures in preparation of cell-based products, but not considered substantial manipulations. So this is taken from a paper in 2012, and it's really just to give you a bit of an understanding of where we were in cell-based and gene-based therapies 3 years ago. Certainly, I don't know whether it's entirely clear, perhaps not on that slide actually, but basically up here what we're seeing is that back in 2012, large pharma weren't particularly interested in these therapy types. They were considered to be quite high risk, and the regulatory path and the regulatory expectations in terms of clinical trials weren't entirely clear because there wasn't a great deal of precedent there. Over the last few years, we've seen a lot of acquisitions, particularly last year, take place, so that companies like GSK, like Novartis, are really becoming much more involved in these cell therapies. But still, what's shown here, we're seeing still a lot of academic groups and small medium enterprise particularly involved. And so that's really where I'm focusing my presentation on today is what are some of the avenues and considerations for smaller companies and academic groups for development in Europe. So since the implementation of the ATMP regulation in 2009, really we haven't seen as many approvals as I think a lot of people had hoped in Europe. So as of the end of 2014, we had 4 products approved, and we just had 1 at the start of this year. So I think that the Europeans were hoping that a lot more products would be coming onto the market in this space, and certainly I think that the number of products in development is quite a lot. But the regulators are very keen to help ATMP developers get onto the market purely because of the small numbers of therapies that have reached patients to date. And certainly of those 5 that are approved, I think only 1 of them is actually available to patients at the moment. So it's a little bit of a unfortunate situation for ATMPs at the moment that we'll hope to change. So moving on, well, considering that we haven't got very many products on the market, I thought I'd review what some of the key technical considerations for these products are, because in my role as a consultant, we often get companies coming to us quite late when a lot of the decisions have been made, and perhaps if they had involved either regulators or more external people early on, then some of the problems might have been avoided. So this is just an outline of some of the quality issues or manufacturing issues that we tend to see at the time of scientific advice that you can get from regulators, or also marketing authorization applications. So if you consider that scientific advice would probably be at the stage of a clinical trial, and marketing is obviously much later stage. So if I point out a few of them there, one of the main ones is that often comes up is the acceptability of the potency assay. So the complexity of these products often means that, and actually one of the speakers before was talking about it, that we don't have a full understanding of how these products actually work. And therefore the development of a potency assay to try and justify what dose that you select for your clinical trials can be an incredibly challenging thing, and one that we have often had to think quite creatively just to put together a story to justify to regulators. But that's certainly one that I would encourage anyone who's looking to move into the clinic to discuss with regulators before you get to that point, because it's often a point of quite a lot of contention. And certainly if we then translate that to a marketing authorization stage, if a marker that you have selected quite early in development as your marker of potency does not end up being correlated to biological activity, it's then very difficult to go back and justify why you've selected the dose that you have, because you can't correlate your activity marker to efficacy. Another critical one is the acceptability of comparability data, and at MAA stage, again, the lack of comparability data. So it's generally understood that over the course of your product development pipeline, as the needs of your product increase, you're treating more and more patients, you'll need to make changes to your manufacturing process to accommodate your needs. And that may be increases to try and ensure greater purity of your product, or simply to scale up so that you have more product to treat more patients. What we tend to see is that the companies don't tend to have a plan in place to to plan for what changes that they'll need to make, and then to also have enough characterization data over the course of their development to then say, right, at phase 1 I had this profile, at phase 3 I had this profile, I've had these changes, but I think that their impact is minor because I can say that I can relate it back to my potency, which is so important, and expression of markers and so on. So I think that the key message from this slide is hopefully that you sit down at the start of your development and you create a plan. And everyone knows that nobody sticks to plans, but have these things in mind and at least have an ideal scenario when you set out. Some of the main non-clinical and clinical issues that we find, relevance of animal models is a huge one. I think we've discussed before about immunogenicity don't worry, it's fine, of cell-based products, and trying to justify whether or not you use a homologous animal model with animal cells or an immunocompromised model with human cells, and how relevant that situation is. These are all things that need discussion in your clinical trial documentation and finally your MAA. At the stage of a first-in-man clinical study, things like biodistribution are particularly important. So with your talk before, I know you said you had some animal data there, so you'd be looking at justifying your proposed mechanism of action by having distribution to the intended site of effect, but also having a worst-case scenario. So if for example, you were injecting intramuscularly and getting some sort of systemic distribution to the liver, then you would want to, the regulators would want to know about that at the clinical trial application stage, and that's certainly an early stage clinical trial application component. And of course, tumorigenicity. So things, I think that there's no expectation for your standard tumorigenicity data that you would expect in, say, a small molecule application, but things like karyotype analysis, anchorage-independent growth assays, looking at activation of oncogenic markers. These are the kinds of assays that the regulators expect to provide some sort of assurance that your product isn't likely to be tumorigenic in vivo. Clinically, dose selection, justification, I think it's start low and go up, really. There's lots of general acceptance that non-clinical studies of cell therapies don't really provide much guidance for your clinical dosing. But still, again, you need to make some sort of case in your clinical trial and marketing authorization applications that you have done multiple dose levels and that you've justified that the one that you've selected is the most efficacious or the most safe. And endpoint and comparator selection. Comparator selection is also a particularly tough one because comparing a cell-based or a gene therapy to small molecule therapies, it doesn't often work. So my intention with these slides is not really to raise up more problems and give answers, but just to acknowledge the complexity of these therapies and to essentially say that because of the complexity, there's not a clear-cut answer, and that's why I think that it's so important that companies engage in interactions with regulators quite early in development to try and form a collaborative approach, because it's impossible to have all the answers yourself, and you may as well inform your development as you go along. So with that in mind, what are these pathways? So the Innovation Task Force has been set up by the European Medicines Agency, and it's really a committee that it deals with cutting-edge therapies. And the advantage of, there's a couple of advantages of this procedure. It's mainly for very early stage dialogue. And I think that the key takeaways from this is that it's a free procedure, and it's one of the few procedures that the EMA offers that is a face-to-face meeting. So a lot of the European Medicines Agency procedures are written, and it doesn't provide a great environment perhaps for much brainstorming or direct interaction. So this is a really good procedure, I think, if you're at an early stage of development and you want that face-to-face direct interaction where perhaps you can just brainstorm ideas. Moving on from that, scientific advice is a more formal procedure. It's offered by both the European Medicines Agency and most of the national competent authorities in Europe. And it's a place to really present questions to regulators, and not only present questions but present your company position. So it's not one that you ask a question, they give you an answer. It's you come with a position, you justify that position and hope to get their buy-in to your approach. I'd say for cell-based therapies and gene therapies and tissue engineering products, European Medicines Agency advice is a fantastic forum because most of the top regulators sit there, and they are the ones at the end of the day that assess these products for marketing approval. And I think the final one that I've got there is data certification. So this is a procedure that I'd recommend more for companies that perhaps are looking for outside investment. And what it does, it provides a pre-assessment of data and identifies major problematic issues which may need to be addressed prior to MAA. So it's the only forum that certifies your data and provides a pre-assessment of data before MAA. So what the incentive is that if you get a good report, you can attract some investment. Obviously the disadvantage is if you get a poor report, then that can, I suppose you better stash that in a drawer somewhere. Yeah, no, I'm only joking. But I think that because of that very thing, people are worried. So only 5 applications have been submitted since 2010, and certainly there hasn't been a great deal of uptake of this procedure. Actually, the final one that I was going to say was just about European Orphan Drug Designation. I know I am running short of time, so I'll keep going. But basically, this offers a lot of free procedures and free interactions with the European Medicines Agency. And the main criteria that needs to be met is this prevalence criteria that the indication of interest has less than 5 per 10,000 persons in the EU. So in terms of avenues to increase the speed to market, so this hospital exemption scheme was introduced in the ATMP regulation to really provide an avenue for custom-made, non-routine ATMPs to be manufactured specifically in hospitals under the responsibility of a medical doctor. The hospital exemption, because it's part of this regulation, must be transposed into national law. But because it must be transposed into national law, what we've seen is that a lot of member states have had vastly different interpretations of what is meant by custom made and non-routine. So how many batches is an autologous therapy made over and over again considered to be custom made? And these questions, of course, I think a lot of discussion in the European community about the legitimacy of how it's been implemented. The advantages for a lot of companies is that it provides an avenue for early access to the market. So if you can administer your product in this sort of setting, you can get some early safety data and perhaps get some anecdotal evidence to again drive investment. The disadvantages are the discrepancy in the interpretation of non-routine, less controlled administration of the product, so it's not under good clinical practice, and therefore there's limitations on how much it can contribute to your final efficacy package. So I'm just coming up to my second last slide now. So just in terms of what's coming in this field, in the regulatory space, there's been talk of a specific EU good manufacturing practice for cell-based products. Also a guideline on the requirements for quality documentation concerning ATMPs in clinical trials, a new European Pharmacopoeia chapter on raw materials, And this final one, the forthcoming implementation of the clinical trials regulation next year, which will take in theory next year, which will take clinical trial regulation from a member state specific process to a centrally approved clinical trial process. So just to conclude, ATMPs in Europe are regulated as one of each of these three classes. The main criteria are these. They're incredibly complex products with a number of technical considerations that we're learning by the day. And I think that because of that, the regulators are increasingly helpful and willing to interact with developers to guide your development path in procedures such as the Innovation Task Force scientific advice and through orphan medicinal product designation. And finally, just the hospital exemption scheme is perhaps an option to consider for early access to market. Thank you.